Okay, so we're going to be talking about unknown number 4 in this segment. This is the gram-positive cocci, and we'll be looking at differentiating the Staphylococci from the Streptococci. You'll get 4 cultures of staph, 4 cultures of strep, and then an Enterococcus faecalis. And we'll be taking a look at those organisms. After streaking your blood agar plate, we'll look at the colonies, and also you want to take a look at the cell morphology, because the staphs are quite a bit different in their appearance morphologically in the gram stain than the streps. The streps tend to be very tiny and in chains. The staphs will be more larger and in grape-like clusters. So you've already had your exercise on streaking blood agar. The thing we'll introduce you to this time, which is a little different, is mannitol salt agar. This is a selective and differential media that's useful with the gram-positives. We'll streak it just like you streak the blood plate in 4 quadrants. And what we're going to be looking for is the production of a yellow colony on this particular agar. That will indicate mannitol fermentation or acid production from mannitol. And this medium also contains very high salt levels, which will inhibit many of the organisms that could potentially grow on this media. So we'll be showing you photographs of that reaction on your Moodle site. So again, you'll perform the gram stain and the Catalase test, which you've already been exposed to. And then the other thing that'll be a little bit different with this lab is we're going to look at the Catalase test as being important in separating the staph from the strep. So your Catalase positive organisms will be your Staphylococcus, and we're going to take those organisms that are Catalase positive and we're going to perform the Coagulase test. Coagulase is done using rabbit plasma. We'll have a slide Coagulase test that we'll be demonstrating in another video. And there's also a test where you inoculate the tube of rabbit plasma and incubate that overnight at 37 degrees, and you'll be looking for coagulation or clot formation in the tube. And that will be very important with your differentiation of your staphs. Now with the streps, we're going to be using a medium called bile esculin. It has a very high level of bile in it, which will inhibit many organisms. So the organism has to be able to grow in the presence of 40% bile first. Then it can produce a black pigment in this, this tube, which will be a positive test for esculin hydrolysis. You're also going to be using a phenol red broth. This is just a standard broth that's used to look at different sugar fermentation patterns. You'll have 4 different tubes in this particular lab. One will have lactose in it, another will have sorbitol, another mannitol, and another trehalose. So if the organism produces acid from that particular sugar, it's going to turn the phenol red indicator yellow, which will indicate acid production from that sugar. So that's also going to help you differentiate many of these organisms that are streps. And again, the streps will be Catalase negative, the staphs Catalase positive. Now the other thing that's a little different is the CAMP test. The CAMP test is performed on a blood agar plate, and you're going to be making an inoculation of the blood agar plate with what we just call a CAMP staph, or it's actually a Staph aureus that you're going to pick up culture from a colony, and you're going to streak right down the center of that blood agar plate with that staff using a swab. Then you'll come back with your unknowns and using either a loop or a stick, you'll pick up a culture of your unknown from a tube. You'll pick up an inoculum there, and then you'll streak perpendicular to that staff, coming up really close to it but not touching it. And this is going to create a unique pattern of hemolysis that we will call the camp reaction. You'll have examples of this on your Moodle page as well. Okay, so this is the Catalase test. We're going to be adding a drop of hydrogen peroxide to a glass slide. Then using a wooden stick, we're going to then apply an inoculum from an isolated colony on a plate. And if we see vigorous bubbling, then we'll have a positive reaction.
So for the Coagulase test, you're going to need sterile DI water and rabbit plasma. First, we get our DI water, we put a drop down. And I'm using a sterile wooden applicator stick, taking colony off the blood agar plate, just mixing it into the water. Kind of like you're preparing a gram stain. Then we're going to get just a drop of the rabbit plasma and put it next to it. And here you can mix the two together. And after about 30 seconds, you'll see some clumping. And there's a little bit of clumping going on here. This would be a weak positive. And you can kind of pick up the slide and just rock it back and forth a little bit. So for the camp test, I've got a sterile swab. I just collected my colony, and I'm going to go down the middle. I'm going to go back over my streak just to make sure I have enough inoculum on there. And using a sterile applicator stick, take a colony of my test organism, and I'm going to go perpendicular to the camp streak. Close to the camp streak, but not touching. And that's it. So this is a phenol red broth test. Every broth that you do, you'll inoculate the same way. I'm using a sterile applicator stick, picking up a colony off my plate or out of your slant, and just dunking it right in and stirring around. That's it. For the bioesculin, this is a slant. I pick up a colony, go in, and just streak it. Then we incubate these for 24 hours at 37 degrees Celsius. So after we made our coagulase slide, we're going to do the same thing except inside the tube. We're going to take a colony off our blood agar plate and then put it directly into the rabbit plasma and incubate that for 24 hours. When you take it out of the incubator, if it's positive, it should be a solid clot. And it's incubated at the same as the plates and the tubes at 37 degrees Celsius.